So for those of you that aren't familiar with interleukin-11, it's funny, you go through these panels of interleukins and IL-11 never comes up when you're, running these broad screens. So it's actually a target that's been around for quite a while. A guy named Jack Elias, as well as others years ago, identified IL-11 as a driver of inflammation and fibrosis, but more recently it's been realized as a fibrotic driver. So it's a member of the IL-6 family. The sort of main IL-11 signaling works by binding of interleukin to the IL-11 receptor. This then recruits GP130 to form this high affinity complex. IL-11 can bind either, there's a soluble form, so in the setting of trans signaling, or in cis signaling, which is the standard signaling modalities you can see here, where you bind ligand to the receptor and recruit GP130. And then the typical downstream pathways, the canonical pathways through STAT3, and then the non-canonical pathways through ERK. And our antibody can actually block both of those pathways, as well as the cis and trans signaling. As I mentioned, IL-11's been recognized in a number of fibrotic diseases, and it's really interesting in that there's a lot of crosstalk between interleukin-11 and TGF-beta, PDGF, as well as other fibrotic drivers as it relates to downstream, fibrotic pathogenesis. So, in terms of the clinical data that's out there where IL-11 has been recognized, so it's been shown to be both IL-11 and IL-11 receptor have been shown to be upregulated in a number of disease settings. The cardiac space was actually some of the initial data of late where they show that IL-11 was one of the most highly upregulated genes by RNA-seq in response to TGF-beta. Since that time, some data has been generated in the liver, particularly in hepatic stellate cells, showing that IL-11 is a driver in the setting of NASH fibrosis or other fibrotic diseases, as well as toxicity-induced fibrosis in the liver. Obviously, we're looking at the lung, so IL-11 in particular is upregulated in lung tissue, and I'll show you some data actually standing for IL-11 receptors, showing where it localizes in IPF tissue. It's also been noted in fibrosis associated with systemic sclerosis or scleroderma. And then, more recently, some work in the GI. We're actually looking at some GI models of fibrotic stricture formation. And then there's also some data in cancer, but that's not being pursued as highly, at least at Lassen. So obviously there's quite a bit of animal data out there. Most of this data has been generated using either knockout animals, so IL-11 receptor knockouts, or using murine surrogate antibodies. You can see here in the bottom left. So across a number of different models in the bleomycin model, and I'll show you some data today from our bleomycin study work in kidney models, namely the folate model, in the cardiac setting. So in angiotensin II induced cardiac fibrosis, which I realize is a bit of an artificial model, but you do get sort of transmural fibrosis. And then in the data you can see here, this is actually comparing some preclinical antibodies that target both IL-11 receptor as well as interleukin-11. So you can get similar efficacy by binding, either the ligand or the target or the receptor. We at Lassen have generated antibodies to both. We're focusing mainly on the receptor targeted approach, but I'll show you some data on efficacy with the ligand targeted approach. So what is the role of IL-11 mechanistically in IPF? So there have been some talks today on, I had a nice talk at lunch about some of the open access RNA-seq data that's available. There are a number of different platforms. I'm not the most savvy person in this respect, but there's quite a bit of open source data out there that people more savvy than I are able to mine and actually pull out some really interesting data. These are data from an open source database set up by Naftali Kaminsky, who's a big leader in the IPF field. Essentially, Naftali's lab and a number of other leading researchers in IPF, interstitial lung diseases, as well as scleroderma have put together this IPF cell atlas. It's very user-friendly. You can go in and look at your target of interest, assess, you can look at specific cell types where your target may be expressed, look across different disease areas, and there actually is links to some of the primary data. So here, in terms of IL-11, just in looking at the various, cell types or stromal cells, epithelial cells, as well as immune cells, we know that IL-11 receptor is highly upregulators highly expressed in fibroblasts. So in mesenchymal or stromal cells, it's increased in myofibroblasts, particularly in IPF tissue, and I'll show some of that data. It's also been shown in the epithelial space, particularly in some newly identified cells, aberrant basaloid. So aberrant basaloid, or a subtype of epithelial cells that are believed to be cells that drive leading edge progression of fibrosis from fibrotic foci, or the fibrotic centers in the lung, into the viable lung parenchyma. And based on some of the work that we've done, or particularly work that's been done by our collaborator Corey Hogaboom up at Cedars-Sinai, it looks like IL-11 receptor is highly expressed in these cell types. And then some of the data in immune cells. So get over here. Particularly the role of IL-11, or expression of IL-11 in monocytes and macrophages as far as myeloid-derived cells. And this has been an area of particular interest to us because there seems to be some specific crosstalk between stromal cells, or fibroblasts, and macrophages in terms of the IL-11 signaling that's occurring as it relates to fibrotic pathogenesis. We've run a number of single-cell assays, and I appreciate the fact that a number of people today have discussed some of their negative data. I agree that it's a shame that we don't discuss more negative data, because we can move forward more collectively or better as a field if we would do this. But so what we found is that that a lot of the IL-11 biology is captured better in these multicellular systems. So we've used systems like the precision cut lung tissue, other multicellular systems, generated organoids, things like that, where we can actually look more at crosstalk between these cells as they interact with each other as it relates to IL-11. And I'll show you some interesting data from Cedars-Sinai when they took dissociated cells from the lung and looked directly at IL-11 as compared to some of the single cell assays. So this is that data. So just looking at the panel over on your left, so this is just staining for IL-11 receptor in fibrotic foci from IPF lung. So what you can see is that IL-11 receptor stains epithelial cells at this leading edge, and what we believe is that IL-11 may be driving, and there's a whole cytokine milieu that we're exploring here to see what other factors are interacting outside of TGF-beta, PDGF, and some of the usual suspects. But based on the receptor expression, we believe that IL-11 may be driving the expansion, or either the epithelial cell death that leads to fibrotic expansion as it relates to progression of fibrosis into the viable lung. In support of this, you can see here, so the data in the middle panel are just normal. So these are cells that were dissociated from human IPF lung. Corey has done a lot of work screening different donors. As you would imagine, there are donor-specific differences, and he doesn't see the same thing in response to IL-11. A lot of times when he goes back, he finds that maybe those patients had more of an emphysematous phenotype as opposed to a fibrotic phenotype. But when he takes these cells, he dissociates them. He has fibroblasts, epithelial cells. There are resident immune cells in this tissue. What he found, as compared to just stimulating cells in monoculture with IL-11, where you do get fibrotic changes, but you don't see as robust of a response, he got this dramatic outgrowth of fibroblast-like cells. So you can see here, you go from sort of a heterogeneous cell population to a typical fibroblast morphology, where you have spindle-shaped cells that grow in whorls. I don't have the data here, but we see robust increases in collagen in these same cultures, so collagen release, supportive of a role of IL-11 as it relates to progression of IPF. And we really think the biology that's going on here is central to how IL-11 drives fibrosis in the lung and likely in other organs. To sort of distill all that down to a mechanism, so what we know about IL-11 is that it's released in response to tissue injury. It's among a number of the factors that are released, as you can see here, CTGF, PDGF, TGF-beta in large part. TGF-beta does drive large amounts of IL-11 release from cells, consistent with TGF-beta being a major fibrotic driver and IL-11 being involved in this biology. In response to IL-11 release, there seems to be crosstalk between myeloid-derived cells, or monocytes, macrophages, and stromal cells that leads to the expansion or outgrowth or transformation of these cells into fibrotic cells that produce extracellular matrix, collagens 1, 3, as well as other collagens as it relates to tissue fibrosis in general. And so I'll show you some data today where if we intervene with an IL-11 receptor blocking antibody, we can abrogate this response in rodents as well as in some translational models. So this just kind of summarizes. So we do have a clinical phase asset, LASN-01. This is a first-in-class IL-11 receptor blocker. We're currently in the latter stages of our phase 1 trial in healthy volunteers. And as I'll show you today, so this antibody has picomolar potency. We've characterized this potency across various cell lines as well as with primary cells. As I said, it's a potent blocker of both the canonical and the non-canonical IL-11 signaling pathway. I'll show you some data in terms of its anti-fibrotic activity across the animal models as well as in precision cut lung from both the normal setting in response to stimulation with the anabios tissues as well as in IPF patient-derived tissues, and we've worked with a number of outside collaborators for that. So far, based on all of our assessments, it's a very safe molecule. It's highly developable. We can produce this in high amounts, consistent with a well-behaved antibody. And then, as I said, we're in it's a clinical stage program as it stands right now, and we're planning on moving into IPF patients later this year and also exploring some other indications where tissue fibrosis plays a major role driven by IL-11. We've tried to take a multi-pronged approach in terms of moving the program forward. So as I mentioned, typical of a program like this, we have a number of in vitro assays, screening assays, potency-based assays in primary cells and cell lines. I almost feel like this should move to the other side in terms of importance, in terms of the translational assays. We've really focused heavily on these translational assays in terms of understanding this multicellular effect that we see with IL-11 and really in understanding the potential for IL-11 blockade in fibrosis. As I said, this involves healthy tissues as well as fibrotic lung tissues. We've also done some work with the folks from CureLine in procuring skin. This was used, one, to assess efficacy in response to stimulation in normal skin and how we can block that, as well as to develop a biomarker strategy that we're employing in our phase 1 trials. And then we've run a number of different animal models, and I'll walk you through some of that data. The most interesting, I guess, at least to me, this humanized mouse IPF model that's run by Corey's group up at Sinai, where we look at human IPF cells in a mouse. So just a snapshot of our signaling data. So as I mentioned, these are picomolar antibodies that inhibit both phosphostat as well as phospho-ERK. These are high affinity antibodies. They're a fully human IgG4. Engineered to prevent chain swapping, and as the data show, they're potent inhibitors. Obviously it's important to look at primary cells. The data that I'll show you today, so we have our lead antibody, which is a human antibody targeting the IL-11 receptor that we're moving forward in the clinic, but we have a number of surrogate mouse antibodies that we use in our rodent studies. The human antibody cross-reacts with human in cyno, but not with mouse, so we can't use that in those rodent studies outside of the humanized IPF model. So it was important for us to assess potency in rodent cells. So these are dissociated lung cells. So essentially the same process that Corey used, excuse me, to dissociate his human IPF lung, we used in mice, culture these and looked at phosphostat. And what you can see here is, this is a simple nuclear translocation assay. So we also have data biochemically using ELISAs, but you can see a nice inhibition curve where, once again, these antibodies are, the murine antibodies, excuse me, are picomolar in affinity, similar to what we're seeing with the human antibodies. So because we also want to look at human tissues, so these are some cells from IPF patients that we've sourced from Lonza. We've spent a lot of time screening various donors for responsiveness to TGF-beta as well as other agonists and confirming the effects of LASN-01. And what you can see here, so these are representative images of just a vehicle collagen expression in a vehicle-treated cells, donor cells. In response to TGF-beta, you can see that there's increased collagen. And then in the LASN-01 treated cells, you can see that there's a dramatic reduction consistent with the dose response that you see here. So we have confirmed that there are very robust increases in IL-11 release in these cultures, consistent with the ability of LASN-01 to block fibrosis. So moving on to the precision cut lung tissues. So these are the tissues from Anabios. I am not showing the IL-11 stimulated data here, but we have similar data where we treat these tissues with interleukin-11 and they can actually elicit similar changes. Excuse me. Sorry, my wife's had a cold and I'm trying not to get it from her, but what you can see is that when we stimulate with TGF-beta, we see robust increases in procollagen as well as TIMP-1. So procollagen being the propeptide of collagen-1 that's released as collagen-1 is secreted. TIMP-1, a biomarker or a marker of collagen as well. And then when we treat with LAS101, you can see similar to the cell data at 3 and 10 microgram per mil, we see nice dose-dependent reductions in release of these collagen or these fibrotic markers. So more relevant to human disease. So these are some studies that were done by Nordic Bioscience. So Nordic, this is a company that spends a lot of their time on extracellular matrix. So they're looking at collagen neoepitopes as potential circulating markers. So the reason for doing these studies was twofold. One, we wanted to confirm that we were seeing efficacy or antifibrotic efficacy in human disease tissues. We also wanted to explore potential biomarkers that we could employ in the clinic in our IPF patient studies. The data I'm showing here for ProC6, which is basically procollagen. So as the collagen is secreted and laid down and cross-linked, these peptides are cleaved off and are circulating, and we can look at those in the blood. These studies were done in culture as a screen to understand which collagens were moving. But what you can see is just media alone, so 100%. Similar effect with isotype control. When we treated these cells. So once again, this is no stimulation. These are actively fibrotic tissues, and we show that we can sort of dose-dependently sort of seeing a flat effect at the 3 to 30 microgram per mil concentration of LAS101. We can inhibit fibrosis in these tissues. We've done more recent studies that actually have statistical significance. As you would imagine, so this is data from multiple donors, multiple regions per donor to really try and give us as broad as we can an idea of the efficacy of LAS101, without just picking a single donor and kind of rerunning the same assay over and over again. But I will say that, one of the caveats with precision cut tissue, and I've talked to a number of friends in the field, is these are variable assays, and you do need to run them multiple times and try and screen a number of different donors to be sure that the effects that you're seeing are real and reproducible. But either way, we see a nice effect here, as we've seen with other markers. The data are in a good agreement with what we've done in-house in terms of the TGF-beta stimulated collagen expression. So in terms of our animal models, one of the typical go-to models is bleomycin. I'm sure a number of you are familiar with bleomycin. This is a cytotoxic agent that when you instill it in the lungs, induces fibrosis in rodents. These are data from 2 different studies. And this first study was one of our preliminary studies, as you can see here, fairly high dose, 15 mg per kg, 3 times a week. But this is comparing an anti-IL-11 blocker versus an antibody that binds the ligand, IL-11. And as I mentioned, we see equivalent efficacy between these 2 antibodies when we target that pathway. We compared to pirfenidone, which was the first approved antifibrotic in IPF. And then the data here just show a saline control. These are all therapeutic studies. So the light gray bar shows fibrosis at day 7. This is the time point when we initiated dosing. Black bar is the progression of fibrosis from that time onward. So really, we were able to halt the further increase in fibrosis in these animals from the time of treatment onward. We've done some additional dose response studies. You can see that same sort of time course here. And then we looked at lower doses, 3 and 10 mg per kg, dosed twice weekly. We see a similar effect. So really kind of blocking the fibrotic response after we initiated dosing. Obviously, we wanted to look at other models. So these are some data from a bleomycin-induced skin fibrosis model. So similar to instilling in the lung, you actually do a subcutaneous injection of bleomycin and you get pretty advanced dermal thickening and dermal fibrosis. You can see those data here. So vehicle animal where you see normal epidermal collagen, subcutaneous fat, and associated muscle, dramatic thickening of that epidermis in the bleomycin-treated animals, a reduction in the anti-IL-11 antibody-treated group at 3 mg per kg, and then here we compared to a TGF-beta blocker. So 1D11 is a widely used TGF-beta receptor antibody. The other thing to note is that there was a maintenance of the fat layer, which is something that you typically look at in terms of another readout of efficacy in these animals. You don't lose that entire fat layer, as you see here in the bleomycin-treated animals. In terms of the quantitative data, you can see the increase in dermal thickness, the reduction with the IL-11 receptor antibody that is consistent with what we see with an anti-TGF-beta approach. And then this is just another readout looking at dermal collagen volume fraction. So this is a computer-based densitometry measurement. We typically like to see at least 2 readouts of fibrosis in any of our models. So I guess more germane to the translational theme of the conference, this is a model that I mentioned that is run by Corey Hogeboom. This is a model that was originally developed by Craig Hankey. And it's so essentially you take immunodeficient mice. It's a typical 6-week model that Corey runs. He does an initial sort of spritz, as it were, of bleomycin, and really this isn't enough to cause a lot of fibrosis in these animals, but what it does is it primes the lung so that these cells can engraft. Corey then takes human IPF tissue, he dissociates that into this mixed cell population. So these are the same cells that I showed you before that IL-11 induced fibroblast outgrowth in. He injects those cells via the tail vein. Those cells then migrate to an engraft in the lung and generate a phenotype that's very similar to what you see in human IPF, at least as far as these animal models can go. You can see here, this is collagen deposition data by hydroxyproline. So a naive animal, just really getting no cells, so just normal lung. In the animals that received the cell injection, you see increased fibrosis, and then LAS101, once again, had a marked reduction in that fibrosis. Corey's typical positive control is nintedanib, so this is the second approved drug for IPF. It's a multi-kinase inhibitor, but suffice to say we had a superior reduction relative to nintedanib in this study. The data below essentially just show body weight, so showing it's a clean antibody. Really, we saw continued body weight gain in the LAS101 treated animals, whereas you can see here there was either no body weight gain or actually decline, which is consistent with what's been seen with nintedanib in preclinical studies. These are just some representative images to show you what the pathology looks like. So here, just normal naive lung parenchyma. Here you can see the vehicle-treated animals. There's significant remodeling of the lung, increased fibrosis, as you can see with the blue staining. There's inflammation. In the anti-IL-11 treated animals, you can see a dramatic reduction, and then the nintedanib is reduced, but somewhere in between anti-IL-11 receptor blocker and the vehicle-treated controls. So we're repeating these studies. Obviously, as I said, Corey's got more donors. We want to make sure these are real. We want to make sure this is conserved across multiple donors when running the same model and doing some other dose response work to understand that as it relates to our clinical program. So overall, this is just a summary of all the different models that we've run, or a snapshot that we're seeing efficacy across multiple bleomycin models, dose responses, whether run prophylactically or therapeutically, humanized mouse IPF efficacy. We're seeing good effects in the TGF-beta stimulated precision cut lung tissue model, as well as in tissues from IPF patients. As I mentioned, we do have human skin data where we're looking at multiple endpoints, looking at collagen release. We're looking at PCR-based endpoints. We have a collaboration with an academic in Germany where we're looking at scleroderma disease skin and actually taking an RNA-seq approach to understand what sort of profile there is in these patients as it relates to fibrosis, potentially in the setting of scleroderma, independent from IPF, and just thinking about other indications that we could move this antibody forward for. Obviously, we're a small company. We can't do everything, but just want to understand the conservation of biology. So just to summarize, hopefully I've been able to convince you that LAS101 blocks signaling in the canonical and non-canonical pathways. I think our mouse anti-IL-11 data demonstrate good efficacy in inflammation. Fibrosis, I didn't show the inflammation data, but we see good reductions in neutrophils and in macrophages in those bleomycin mice. We see consistent inhibition of PCLS endpoints. And then really, we believe IL-11 is a prominent fibroinflammatory target. And so we're moving forward for IPF in that indication.